Chapter 13, Flow Cytometry. So we'll go over what a flow cytometry basically is, how it works, what are the clinical applications of it, and basically just about immunoassay very briefly. So flow cytometry is a way for us to analyze cells and identify them individually by an automated method that puts the cells into a suspension, into a liquid. So an antibody with a fluorochrome attached to it will attach to the cell, which will be the antigen. So the monoclonal antibody will attach, and the monoclonal antibody has a particular fluorochrome attached to it. So it can analyze cells, tens of thousands of cells in seconds, extremely fast. And usually what the antibodies, the monoclonal antibodies, are to a CD marker. That's what we're using to identify the cells. So this is important, of course, for leukemias and lymphomas. It actually started out with HIV, where we were trying to identify the CD4 cells, the T helper cells, which, that's what helped us to determine what was actually going on in HIV. Because in HIV, the virus actually gets into the T helper cells, the CD4 cells, which, that's what they should do. And it continues growing in those cells. So the person initially gets antibodies, but what will happen in the later stages of HIV, when it gets to the true disease state, it will actually kill the T cells, the T helper cells, which hence make you immunodeficient, hence the HIV name. This is actually a guide too for treatment, so they can actually monitor how many CD4 cells you got, and there's a certain number that they're looking at to determine if you have HIV or not, and we'll discuss that when we get to that chapter. So anyway, that's one of the things. Another one is for any immunodeficiency diseases, so we can look at the different types of T cells and B cells to do that. And it's good for stem cell transplants, because we'll actually enumerate how many stem cells are actually in the stem cell transplant, and the CD marker for stem cells is CD34. So we can use it to detect HLA antibodies, which makes it really easy. We can use it to determine the DNA content in tumor cells. So a whole bunch of different things that can be done. So here's a picture of the instrumentation of it. You can see the fluidics. You can see the laser here. Now here's the air pressure. Here's the cell suspension. It comes out and it goes through one, and then it has a sheath fluid. In other words, what that sheath fluid is doing is it's actually allowing the cells to go in this particular case, a downward fashion, one at a time. It's sort of like, your sink, and on the outer edge of the water of the sink is the water, and in this dead center is where the cells are. So it keeps the cells going in one direction. So we have that, so it goes through, and then here's the laser, and it has lenses, and you can see it will measure the light coming off of the side. They call that side scatter. And there is also a forward scatter. So it's looking at that. So as the goes through the laser, it hits it with a certain wavelength, and then it excites the fluorochrome, then emits a different wavelength. And this particular one is showing you it's got 4 different ones here. So it gets all that information and plots nice, pretty little graphs for you, so you can figure it out. So this is just looking at, one particular fluorochrome here, the flow intensity of it, and the actual counts. So it's looking at basically the whole width of it. That's what those little bars are up there. So it's saying all these in this area and that little blue area, these are blah, blah cell. Basically is what it's telling you that. So what it does is it gets into what's called a dot plot system. And we can actually sort of see that on our CBC analyzer, but we don't have fluorochromes in this particular case. We're looking at the size of the cells and the components of the cells. It's part of the technology that they can do. They look at the granules, they look at the nucleus, they look at how much cytoplasm, all that together. They get all this information, and then they plot a dot as to what all this information says. So in this particular one that you're looking at, this dual plot, is there's an x-axis and a y-axis, okay? And so what they're getting is they're getting lysed whole blood, so there's no red cells, you know, so that's why they're down here. And it's actually showing you where the red cells are, where the lymphocytes are, where the monocytes. And then in the red is the neutrophils. So what they've done is they've put a gate around it, like a fence. They call it gating. That's the term they use for that. So they're surrounding, we'll say. So they're saying everything in this gated area is neutrophils. So that's what the gating is doing. So we can say, this is what we're after. I want you to enumerate those and tell me what that is. Okay? Now, you can also gate cells in a quadrant fashion here. This is particularly looking at 4 different ones here. So you can see on each axis is some fluorescent positive and some fluorescent negative cells. So that's, you know, the example we're looking at is determining, especially here in the bottom one, is a dual parameter dot plot, and we're looking at CD4 cells as opposed to CD3 cells. So it's telling us how many that it got, and you can actually see it's come up with the numbers in this. So we're after the CD4 cells, like HIV. So it gives us an actual number. It will do that for us, the flow cytometer itself. Okay, so the sample we use, normally we use whole blood, and it's collected in EDTA tubes, a laboratory tube, and you need to process it pretty soon after you get it. You store it at room temperature because white cells do not like going to refrigeration temperatures. Make sure you mix it well, and then there's a whole bunch of stuff you got to add to it, which we're not really going to go into that too much. You can use bone marrow and all that. One of the biggest things is the red cells cause problems, so they lyse it and just leave the white cells. I know you think, well, why don't they just use Buffy coat? Well, there's still red cells in there. So that's what they do. You can use tissues. That's usually tissues grow into a tissue culture medium, you know. And what they'll do is they sort of digest it to a certain point, we'll say. And we'll have antibodies, monoclonal antibodies, that'll be added to it so that we can identify the cells again. Okay, now there's a video that you can watch that will explain it a lot better. You know, it's got a lot of more video kind of stuff explaining it, so it'll be a lot better that way. So make sure you look at that video. So anyway, immunoassays. You know, if you're going to automate it, you want it, you know, accurate, precise. It reduces the errors. You know, fewer people. So, you know, you save, you know, it's better identification, barcoding. It's like, you know, this is common sense stuff for us. In this chapter, this is sort of thrown in here, but it's just so you'll know what a batch analyzer is as opposed to a random access analyzer, because this is basically any kind of chemistry analyzer also. So batch analyzers can examine multiple samples, but it's only like all the glucoses and then all the BUNs, you know, that kind of deal. A random access analyzer can do multiple things. Say, you know, you got a patient that needs electrolytes, sodium, potassium chloride, CO2, BUN, creatinine, glucose, all this stuff. What it will do on patient A, it'll run all that stuff, and then it'll go to the next patient. So you'll get all the results for all the tests you required on that one patient at the same time, or very close to the same time, as opposed to running all the glucoses and then running all the BUNs and all the whatevers. So most of the instruments now are random access analyzers. So automated stuff, put sample in, it adds the reagents, it mixes the stuff, it incubates it, does a reaction, detects it, calculates it, reports it out. So what we have to do for immunoassay automation, like the Vista, if it was running, is that, you know, all this is regulated by CLIA, Clinical Laboratory Improvement Act. And what it says is that we have to validate. Now, we got to say for sure that it's accurate, it's precise, it's analytically sensitive, and it's analytically specific. And what are the interfering substances? What is the linear range? What is the normal range? You know, and usually the manufacturers of the companies that make these instruments do all that for you. They do all that validation study for you when they set the instrument up. You know, because it is very time consuming. to do. It's a whole bunch of stuff you got to go through. So if you didn't know this already, accuracy tests ability to measure what it accurately should say. I mean, to the true result. Precision is to be able to reproduce it over and over again. Analytical sensitivity is the lowest measurable amount of an analyte. In other words, for like, say, glucose, which isn't a great example. To say glucose, I want it to measure down to 2. I mean, sort of immaterial when it gets up, but anyway, that's beside the point. So that's the analytical sensitivity. The analytical specificity is an assay's ability to generate a negative result when the analyte is not present. In other words, if there was no glucose in this particular example, in this patient, then it should say 0. In other words, we don't want, stuff causing problems. We want to be specific for what we are looking for. And then the reportable range is that will give us a result for the specimen. Reference interval, another name is normal range. This is what we call in healthy individuals. So it's tough to come out with a normal range. Negative, positive's great, that kind of deal. But what's the normal range for glucose? What's the normal range for BUN? That kind of stuff. So and for your population. So there's a lot of work that has to go into that to help us identify what is truly a normal range. I mean, you got to look at age, sex, race. There's a lot of things that have to be involved. So you got to go by your patient population. So summary, what does a flow cell do? Identifies and enumerates all kinds of cell populations in a fluid. So each particle goes through with hydrodynamic focusing, if you remember that from hematology. So it goes through a laser light and produces characteristic side scatter, forward scatter patterns. We can use fluorescence also to help us identify things. So make sure you look at that video. It's very good. There's a side scatter, which is 90 degrees, which tells about the granules, the complexity that's inside of the cell. And then they plot it on a graph, X and Y axis. You can get single parameter or dual parameter, so you can look at multiple things. In flow cytometry, we sort of put a fence around it. We gate it. That's a term we're using, gating. So it tells us what particular cells we're looking at so it can enumerate it better. So flow cytometry is very important for immunophenotyping for lymphoid and myeloid populations, looking at the CD markers. So they have the monoclonal antibody with the fluorochrome, looking for that particular CD marker, run it through a flow cytometer, and you can enumerate how many of that CD marker are present. Automated immunoassay analyzers, the validation, what accuracy and precision, what analytical sensitivity is, specificity, what a reportable range, in other words, it's also called linearity a lot of times, and the reference interval. So this is a really short chapter. I didn't really go into all the different surface markers and all the junk they do. I mean, there's tons of them in there. I'm not going to ask you that particular stuff because I got to look it up half time. I can't remember. I mean, it's sort of getting pretty common to do them now, but like CD4 helper cells, that's a biggie, of course. So it's got all the different analyzers. I'm not going to ask you that. I can't remember all that mess either. So look at the summary, of course, and look at the review questions. If you have any issues or questions, let me know. Text, call, email. I'll see you in lab.